Case Western Reserve University's Institute for the Science of Origins proudly presents the Origin Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origin Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. Tonight, it is my pleasure to introduce Cynthia Beall, a distinguished university professor and S. Idyll Pyle Professor of Anthropology at Case Western Reserve University. Professor Beall is a member of the U.S. National Academy of Sciences, the American Philosophical Society, and the American Academy of Arts and Sciences. She's a physical anthropologist whose research focuses on human adaptation to high-altitude hypoxia, particularly the different patterns of adaptation exhibited by Andean, Tibetan, and East African Highlanders. Please join me tonight in welcoming her to talk about tracing evolution, where modern human variation comes from and why.
Up here on the screen, you've got a half dozen specimens of ancestral species, Homo erectus. And they all lived about 1.8 million years ago, and in the same place in what is now the country of Georgia. And one of the first things that probably sticks out at you is the differences in how much their face projects in front, how big the bones, the brow ridges are above their eyes. You know, some of them have almost points at the top of their heads. So here we have 5 individuals who just happen to be preserved and just happen to have been excavated and who lived 1.8 million years ago, and we see variation. So rather than simply saying, oh good, we see variation, let's catalog it, we'd like to move on and be more scientific and say, why do people vary? And in fact, human variation is abundant in living people as well as in fossil specimens. Some of it is visible, as in the case of these 3 children. We can see differences in hair color and skin color. Some of it is invisible, as in the case of the ABO blood group. The ABO blood group is something that we share with primate relatives. So we've got it, it's visible, it's invisible. Importantly, we can measure human variation. And our aim, or my aim as a biological anthropologist or physical anthropologist, is to understand why we vary. Just to give you an example of something you may have never thought about that varies, let me ask everyone to clasp your hands like this. Okay, what thumb is on top? Yeah, okay. Right, left, yes. Human variation. I have no idea why, but that is always a good, but that is always a good one to get people to thinking about variation. So at the theoretical level, we vary for 2 reasons. One is that each of us in this room, each person on the earth, has a different life history. We've been exposed to different environments, we've lived under different conditions, and those social and cultural and environmental factors influence our biology. And also, each one of us has a different genetic heritage. So for example, on the lower left here, the young baby is going to grow up and be exposed to a very different environment, and I don't know if that's a his or a her, but his or her grandmother on the right, and therefore their biologies may differ as adults. And on the right, we have a modern day human facing off against a modern day human dressed up as a Neanderthal. Illustrating that we may have genetic heritage from our Neanderthal ancestors. To give you an example of life history as a source of variation, let's take height. Often in a population, this is a population of young men drafted into the US Army over, well, I guess they were volunteers in 1710, but these are young Army recruits, let's call them that, in the US. And you can see that over time, between 1710 and about 1790, average height increased. It increased a little more up to 1830, and then it plunged from about 5'8.5" down to about 5'6.5" between 1830 and 1870. Now that is much too fast for genetic factors to be the explanation, and height variation over time like this is attributed to differences in the environment, differences in the kind of food that people eat, the kinds of work they do, and particularly the kinds of infections. So in this case, between 1830 and 1870 or so, many people were moving to the cities. There was a big rural urban migration, and life was harder in the cities. The sanitation wasn't very good, people were crowded, and there was more infectious disease. And you can see a big difference in adult height, young adult male height. Then between 1870 and 1970, see a steady increase in height, up from about 5'6.5" to almost 5'10". And this is attributed, again, it's too short a time to be attributed to genetic factors or changes, but it's attributed to improved sanitation, fewer oral fecal diseases, improved nutrition, more fruits and vegetables, and better health overall. So here is one example where we see variation in height over a period of decades and centuries, and we can attribute it to variations in individual life histories. Genetic inheritance is another source of variation, and my example here is called the Inuit paradox. Here we have a picture of a community of Greenland Inuit, and they're crowded around a beluga whale tail, and they're all eager to help themselves to a piece of whale meat. And the Inuit, living where they do in Greenland and northern Canada, do not eat much fruits and vegetables. They eat lots of sea mammals and fish, and their diets are very high in fat. Yet, they don't have heart disease. They have very low frequency of heart disease. And this has been interpreted as, well, wait, this doesn't make sense on the basis of what we know about risk factors for heart disease in our own country. And recently, examining the genetics of the Inuit in comparison to nearby populations, we've learned that the Inuit, in fact, have unique genetic variations in metabolic pathways for digesting fat. And that variation in their metabolic pathway keeps their cholesterol level low and their insulin levels low, even though their diet is high in fat. So now we've got a couple of examples of sources of variation. Another well-known source of variation is ancient inheritance. In this case, there's a gene called MC1R that, in the case of the blonde and the black-haired mice, determines the difference in their coat colors. That same gene, variation at that same genetic locus, influences whether or not a woolly mammoth is a red-headed woolly mammoth or a brown-haired woolly mammoth. And similarly, variants at that same gene influence whether or not the Neanderthal has red head or a modern person has a red head. And this gene has been around for hundreds of millions of years because MC1R and its variants influence color in mammals, birds, and reptiles. So in the case of the Inuit, we had what we think is relatively recent, in the last few thousand years. Here we've got genetic heritage of on the order of millions of years. Migration from other populations is another source of variation. This map shows actually it's from 2016, it's from the BBC, and you'll remember that this was the year that hundreds and thousands of migrants were streaming in from the Mideast into Europe, and many had a destination as Germany. And one of the things that many Germans were saying to themselves is, we don't want all of these people. We have our genetic heritage to think of. So we can ask the question, well, what is the genetic heritage of Germans? And it's very interesting because we know something about it from using ancient DNA as well as modern DNA. For maybe at least 50,000 years, there was a population of Homo sapiens living in Europe. Somewhere between 19 and 14,000 years ago, there was a migration into Europe from the Middle East. These were people who were hunters and gatherers. They didn't have they weren't farmers yet, but they did migrate into Europe. Okay, so there's one source of new variants coming from the Middle East into Europe. About 9,000 years ago, a group of farmers migrated into Europe from Turkey. Second source of genetic variation. About 5,000 years ago, herders from Russia migrated into Europe. So the modern European population is a mixture of a very old population and these various migrant populations, all bringing with them slightly different genetic variations. Now, this is a part of my page from 23andMe. Have any of you done your 23andMe genotypes? Yeah, okay. Before we go any further, yes, I know this is recreational genetics, but still, it's fun, and it'll illustrate my point. And that is, again, where does genetic variation come from? So this is my mitochondrial haplotype is one of the variants of U up here. And mitochondrial genes are inherited via the maternal line. And the oldest mitochondrial variant emerged in East Africa. It's labeled L there. So this is the original. And then that mitochondrial population of women, only women, transmitted the L variants to their offspring. Their daughters, only their daughters transmitted that variant to their daughters, and so forth. And over time there were mutations and there were evolutionary branches, and you can see the kind of the temporal and geographic order. L, one of the lineages was L3. Further north, another lineage, N, R, and U. So this is kind of my mitochondrial inheritance over the past 20,000 years. What about my Neanderthal history? Neanderthals lived between 130 and 30,000 years ago in Europe. And 23andMe will also give you the number of Neanderthal variants that you have. I have 292, for what that's worth, out of millions, but so I've got some Neanderthal variants. One of them has to do with whether or not I sneeze after eating dark chocolate. I do not sneeze after eating dark chocolate, and apparently neither did my Neanderthal ancestors. And then finally we can look at a more recent, again this is my 23andMe report, more recent evidence in the past few centuries of the geographic origins of my family tree. And it's plotted around the world here, where you can see that I am 99.9% European, 0.1% sub-Saharan African, and within Europe I have all sorts of various British, Irish, general, Scandinavian, and so forth. There's a lot more information for Europe, a lot more genetic information, so it's much easier to make inferences about multiple populations as being part of my ancestry. In the case of Africa, there's not we just don't have very much data from West Africa. So my African ancestry is depicted in a really big blob here of African ancestry. So these are ways that we can find out genetic sources of inheritance. We've talked about life history, and we've talked about genetic differences. And now it's time for questions.
Wouldn't the single biggest cause simply be the environment, like with your example with the Inuits? If they did not have a disease that would allow them to process fatty foods and keep the insulin and cholesterol levels low, they would die. When you're living in a cold environment, you would be basically eating animals which also had protective layers of fat to stay warm.
The situation that you're talking about is what I'll talk about next, which is that we evolve and adapt to certain aspects of our environment. So that's going to be another source of variation.
I had read where people that have red hair need to be very careful about anesthetics because they need more anesthetic in a surgery or something because they experience more pain. Do we know anything whether that would have been also with Neanderthal?
Boy, I don't know. Is there a surgeon in the audience? Anyone know about pain and redheads?
I'm a retired dentist, but at Metro Health Medical Center, we treated developmentally disabled kids for a long time, and the anesthesiologists were aware that it required sometimes up to 25% more anesthetic to get acceptable anesthesia.
In redheads?
In redheads. It was almost a marker.
How about that? Well, thank you. We've all learned something.
The specimens you showed us from Georgia, were those contemporary or were those over time?
No, they all lived there at the same time. Well, the time is 1.8 million years, so and that's plus or minus, on the basis of our dating techniques. So I don't know that they were neighbors.
But it wouldn't be that species changing over several 100,000 years, as far as you know.
Yeah, that's a good question.
Thank you for joining us. You've been watching Dr. Cynthia Beall discussing how both life history and genetics contribute to human variation. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Beall will focus on how natural selection favors the evolution of lighter skin color for humans living at higher latitudes. Now, back to the talk.
To pick up on one of the questions, evolution and adaptation to different environments is, in fact, another source of variation. And you've heard before in this lecture series that evolution and adaptation results in well-adapted organisms. And the picture up here is of a blonde rock pocket mouse living in the southwest US. And you can see that this cute little guy is blonde and is on black lava. Now, the main predator of rock pocket this mouse, is our owls. Owls are visual predators. So this little blonde guy is going to get picked up immediately, right? However, if he were living in the background of this picture where there's grass and sand, he would blend right in. And in fact, in 2 or 3 different areas in the southwest of the United States, we have a beautiful example of evolution and adaptation where we've got black lava flows where mice live next to sandy, grassy areas where mice live. And in both areas, we see that the sandy blonde areas have blonde mice. Rarely are there black mice, and when there are black mice, they're more likely to get eaten. Similarly, when the blonde mice are on the black lava, they're more likely to get eaten. And that, as I showed you, the difference between the two is in coloration is the MC1R gene. So here we have a case where there is variation in the environment, variation in the organisms, and the environment patterns the variation in the organisms in a direction that results in organisms that survive better in their environment. So that's just by way of giving you some background. And now let's examine 2 cases of human adaptation to the environment to illustrate the resulting variation. This is a picture map from NASA illustrating surface temperature. But I'm not going to talk about temperature. I'm going to talk about something else. This is a picture taken by geneticists, or a collage of pictures from geneticist Sarah Tishkoff, who has worked with many, many African populations. And one of the points to take away from this picture is that these people all come from Africa, and there's a very wide range of variation in skin color. And skin color has been a topic of interest for scientists for centuries. The point that I would like to make about skin color is that we can measure it objectively and quantitatively. The set of tiles on the left is called the von Luschan scale, after Dr. von Luschan, who devised it. It's a series of tiles of different colors that he thought matched skin colors, going from 1 to whatever it is, 50-something up there. And so what people used to do is go into the field or go to their neighbors or whatever, and they could match someone's skin color against these tiles. And so they were trying to quantify skin color without and putting it into a bunch of categories. Nowadays, as indicated on the right, we can measure skin color objectively, quantitatively, by measuring skin reflectance. Skin gets its color on the basis of the amount of incident light that's reflected. Darker skin, less light is reflected. Lighter skin, more light is reflected. And we can measure that. And Nina Jablonski, anthropologist from Penn State, is doing it here with a study volunteer from South Africa. And that gives us a nice quantitative trait. It gives us something called melanin index, which is a transformation of light reflectance. And you can see that the melanin index in this case is going from 20 to 120. And she's got 2 populations here. One population is indicated by blue, the blue bars, and the most frequent, and that one goes from roughly 20 to maybe 60 units. The most frequent being maybe 45. And it overlaps with the population in the yellow that ranges from 40 to 120 with an average of around 70. So my point here is that it's quantitative and that we can actually look at a mean, a standard deviation in one population and compare it with a mean and standard deviation from another population. That makes it a quantitative trait. It's also a polygenic trait. It's a single trait, but it's influenced by many genes, each with a relatively small effect. So one's skin color is a result of the alleles, the variants that you have at a whole bunch of genes that are listed here on the right. You've already heard about MC1R and these others, and there are more. But my point here is that skin color is a quantitative trait. It's determined by many genes. We can measure it. Now let's plot skin color against latitude. And you'll see that there's a very smooth gradient. The lightest skin color is represented by a little bit of an off-white, and then we're going into the darkest skin color that is almost black. And you can see that the darkest skin colors are in the tropics. And that moving away from the tropics, we gradually see that skin color gets lighter and lighter and lighter. There's no abrupt fall off between light and dark. Instead, there is a gradient from one point to the other. And we call that a cline of human biological variation. So now we've measured things, we've plotted them against globally. And what do we think accounts for the variation is variation in ultraviolet radiation, which also varies globally in a pattern very similar to skin color. This is UV index for a single day in October, and it ranges from low values in the greens up through very high values of UV in the light purples. And where do you see the two lightest purples? You see them in the tropics. You see them overlapping very much the areas that you saw in the previous plot where was the darkest skin color. Similarly, you see the areas with the lowest UV index overlapping with the areas with the lightest skin color. So what? Well, ultraviolet radiation exerts a biological effect on us, an extremely important one. And the effect it exerts depends on how much ultraviolet light penetrates our skin or not. And you've heard of vitamin D as the sunshine vitamin, and the reason for this is that ultraviolet radiation from the sun initiates the synthesis of vitamin D in the skin. And this diagram from the, I think it's from, I forget where, is the NIH, I believe. It probably says, if I could read. Med movie, all right. It shows that ultraviolet radiation penetrates the skin. Ultraviolet radiation B penetrates and just below the outer layer of the skin, it initiates the synthesis of vitamin D. Further in, ultraviolet radiation A penetrates further, and it can actually penetrate into blood vessels. So the thing that we want and that we need from ultraviolet radiation is vitamin D synthesis. However, so we need enough vitamin D for that, ultraviolet radiation for that to happen. However, too much ultraviolet radiation also has costs. It can cause DNA damage in the cells in which it hits. It can cause the suppression of the immune system. It can actually, ironically, degrade vitamin D. It can also degrade folates. And folates are necessary for DNA replication, for normal growth and development, and so we don't want them to be degraded. Now, the amount of UV radiation that penetrates our skin depends largely on skin pigmentation. So now we want to know then what determines skin pigmentation. And that is a pigment molecule called melanin that we have in our, the outer layers of our skin. And the more melanin granules we have, the darker our skin. The melanin granules are dark, and they absorb relatively more of the ultraviolet radiation. So here you have a cross-section of skin from 3 different people. And on the left, you see virtually no black pigment, no granules of melanin. In the middle, you see some granules of melanin, someone with a slightly darker skin color. And on the right, you see a lot of melanin granules. So this is what gives skin both its color and a way to calibrate how much ultraviolet radiation penetrates into the skin, into the layers where vitamin D is made. Now, UV radiation can be stressful, as I implied. We need enough, but not too much. And so here's a plot of UV radiation with the world divided into 3 stress areas. Zone 1 is the clear area there, kind of between the tropics of Cancer and Capricorn. And these are areas where there is an adequate ultraviolet radiation all year round to make vitamin D. So you're good for vitamin D here. However, that, if you'll recall, is also the area of the highest ultraviolet index. So the potential here is also for the highest DNA damage, the highest degradation of folates and vitamin D. However, this is also an area of high pigmentation, high melanin index. So we can put that together and we can say, oh, the melanin is protective against DNA damage, the damage from ultraviolet radiation, yet enables enough UV radiation to penetrate that vitamin D is synthesized. Move up into zone 2, as indicated by the vertical lines. We're in zone 2, and in this zone, there is not enough ultraviolet radiation to make vitamin D for at least 1 month a year. Here in Cleveland, from about October through March, the sun may shine, but you're not getting UV radiation to make vitamin D. You have to rely on, it's fat stored, so you can rely on your fat stores. When I ask students nowadays in my class, where do we get vitamin D, they say orange juice, because they're used to fortified orange juice with vitamin D. But until very recently, it was us and our UV radiation that determined how much vitamin D we had. This is zone 2. Remember, this is a transition zone for skin color. And then zone 3, the squares at the top and the bottom, are where on the average throughout the year, there is not enough ultraviolet radiation to make vitamin D. And what you see in these populations is that they have very, very little pigment. And that enables what UVB there is to penetrate the skin, initiate the synthesis of vitamin D, and which can then be stored in their fat. So they're relying on their fat stores. So we have a global selection gradient which favors different phenotypes. That's the technical term for biological variation at different latitudes and with different ultraviolet stress. In the northern latitudes, or the very southern, we have low ultraviolet radiation, and that selects for light pigment. It reduces, and the light pigment reduces the risk of vitamin D deficiency. It enables the normal function of the skeleton, nervous system, immune function. In areas of high UV radiation around the tropics, we have natural selection for dark pigmentation, which reduces DNA damage reduces immune suppression, reduces destruction of vitamin D and folates. You'll remember from that plot that light pigmentation is found at high latitude both in Europe and in Asia. So the biological trait of light pigmentation and little melanin is found in both continents, but the associated genes are different. So evolution has acted on random mutations, on different random mutations in the two geographic areas. Which does also raise the question of what was the origin of skin color variants. Interestingly, many of the variants that we see today, both for dark skin and for light skin, originated in Africa. In Ethiopia, for example, about half the genes for one particular locus, at one particular gene that influences skin color, about half of them are for light skin color. They're the same as that are found in Scandinavia. Yet all the other genes that they have are genes variants for dark skin. Similarly, the genes for dark skin throughout the world, for example, in southern India, in Melanesia, some of them also come from Africa. So some of our variants for skin color are very, very ancient, both for light and for dark. So global variation in skin color illustrates a continuous gradual variation, explained by continuous gradual variation in the physical environment. And these forearms from around the world illustrate that gradation. Time for questions.
Going back to that chart showing from 1830 to 1870, big dip in height, but rather than just hygiene and sanitation and that, wouldn't that have been the result of two incidents, the Civil War and the influx of immigrants from Europe, specifically Ireland, who were then drafted?
I do not know the source of this particular data set. Certainly, I would be very comfortable adding the war as an environmental factor. Although I'd be a little hard, I guess you'd have to argue that nutrition became poorer and health became poorer throughout the country.
Well, a lot of the Irish were coming over as a result of the potato famine. And there was no draft prior to the Civil War.
I honestly don't know about the Irish. So,
they're a little people, you know.
Hey man, you saw my genealogy there. We're plenty tall. I'm not an example of one, but.
Is there a reason why humans evolved, adapted the way they did to make vitamin D instead of maybe evolving some way of making vitamin D without the UV radiation? Is there a chemistry or physics underlying that?
I think that you're asking, let me think, how do I phrase this? What evolution works on is just what happens to be there. So this is what happened to be there. And I don't know enough to know if that particular pathway was doing something else before we were using it for vitamin D. Do you know? No, okay. So what I would say is that I would hypothesize that those variants were there and they enabled us to synthesize vitamin D. And the reason it was important was that our diets did not have a good source of vitamin D. If you think of the dietary sources of vitamin D that you think of now, it's oily fish, for example. And we don't eat a whole lot of that.
What time course are you talking about for most of these changes? You know, family moves to the Andes or family moves to Africa. How many generations is it later that you start to see perceptible changes in ability to tolerate altitude or sunshine or whatever?
It'll vary depending upon the stress and depending upon the population. For example, for skin color, we hypothesized that our earliest human ancestors were like Ardipithecus and so forth that you've probably heard about from Dr. Haile Selassie, probably still had body hair and pale skin. And it wasn't until we lost our body hair, moved out onto the, into full sunshine, that there would have been selection for light skin. So there we might be talking a couple of million years. We can look in Europe at some ancient DNA, and we can see as recently as 10, 15,000 years ago, we do not yet see light skin color. So it's possible that light skin color evolved in the last 15,000 years. With respect to the Andes and Tibet and people living at high altitudes, the longest they've been living there is around probably, again, same time period, 10, 15,000 years. The next example I'm going to give, however, we know evolved in a lot shorter time, over a few thousand.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Cynthia Beall. Dr. Beall is Distinguished University Professor and Sarah Idyll Pyle Professor of Anthropology at Case Western Reserve University. A member of the National Academy of Sciences, she co-directs the Center for Research on Tibet and is a fellow of the Institute for the Science of Origins. In the second part of our talk, we learned how the need for vitamin D, produced in our skin with the aid of ultraviolet radiation, drives the emergence of lighter skin tones in human populations living far from the equator. In our final segment, Dr. Beall will describe how certain human populations have evolved the ability to digest milk products well after infancy, unlike our Paleolithic ancestors who had no dairy in their diets. Now, back to our talk.
Now for another stress. And this is a stress we create ourselves. Unlike ultraviolet radiation, our diets nowadays rely on domesticated plants and animals. That was not always true. It was really about 10 to 12,000 years ago that we began to domesticate plants and animals. And the red squares on this map indicate areas of early domestication of plants. And you're all familiar with the Fertile Crescent that we all learn about in the third or fourth grade, right? This is where wheat and barley was first domesticated. And it was about 10,000 years ago. Millet was domesticated in other places. Potatoes were domesticated in the Andes. Rice and millet in East Asia. And you can see that this is all over the map, literally. It's not in any nice, neat gradient. It just occurred where it occurred. And agriculture spread quite rapidly. It has real advantages because it can create surplus food that we can then store. The drawing here on the top is a picture of an early grain storage facility, so to speak, from Turkey. So you can grow excess food, you can store it, and you can support more people. The picture on the bottom is by Brooke Thomas, and it's illustrating potatoes in the Andes, and you can see that in the Andes. They grow lots of different kinds of potatoes. So we generally think, oh, agriculture is good. We like agriculture. Health and nutrition, however, were generally poorer after the transition to agriculture. In areas of the world, and we have about 10 or 12 of them, where we can look at skeletal populations of people who are living as hunters and gatherers, and then we can watch other populations later as they were transitioning to domesticated plants and animals, and after the full transition. And one of the things that we see almost uniformly is that height got shorter. we lost height. So people's life histories were more stressful, partly because often when you're eating domesticated plants and animals, you're eating a smaller range of food. You're living in villages, sanitation is poor, more crowded, more disease, and so forth. We also see across the transition the increase in the prevalence of cavities. And on the lower right, you see this molar with a little hole in it. That's from about 8,000 years ago. That's the first example we have of dentistry. I would not like to have been the patient of that dentist who the patient got a little hole drilled in the part that hurt. However, we have regained the height, and we have improved our dentition, and we have adapted to parts of our diet. The plot, or the little plate on the left, you all know from the US government. It tells us that we should be eating roughly equal amounts of fruits and grains, and vegetables and protein, and dairy. And I'm going to focus on dairy. And the pie chart on the right I made from reviewing a series of publications, and it illustrates, using similar colors, the proportion of the Paleolithic diet that is contributed by the same elements as the My Plate figure. You'll notice that there's no dairy. Before we had domesticated plants and animals, adults, or actually children after weaning, did not have dairy products. Just mother's milk was the only source. You also see that we were eating a lot of protein, more than is recommended now. A lot of vegetables and fruits. Rarely did we have grains. They were wild grains when they were. So there's been a big difference in our diets. And we have adapted genetically particularly strongly to one of these dietary changes, and that is the availability of milk. Having domesticated sheep, goats, camels, cows, yak, and so forth, we now have milking animals, and we can drink milk after we're weaned. The earliest evidence that we have for the use of milk dates to about 6,500 years BC. And we can tell this by the residue of proteins on the inside of pottery. Now, just because we had milk available to drink does not mean that we actually drank milk. And that's because milk has a unique sugar called lactose that not everyone can digest. It requires a special enzyme called lactase to break down the lactose so that it can be absorbed. If you don't make that protein, that enzyme to break down lactose, you don't get those calories from the lactose in milk. And you're probably whirring through your mind, is lactose tolerance? Yes, you're right. A trait called lactase persistence is a contrast with a trait called lactose non-persistence. Lactase persistence is also called lactose tolerance, and this is controlled by a single gene. And this is a gene that results in continuing to produce the enzyme lactase throughout your life. There are 2 glasses of milk here. On the left is someone with the genotype for lactose intolerance who drinks a glass of milk, does not digest the lactose. The lactose moves its way along the GI tract, gets used well. The bacteria are very happy to make up the lactose, to use it up, and they build up gas. That causes bloating, gas, and diarrhea in people who are lactose tolerant. On the right, you have a glass of milk consuming full of lactose. being consumed by someone who has one of two genotypes who's lactase persistent. And that individual can make the lactase that breaks down the lactose, and they can then absorb all of the calories in their milk. And they don't get sick from the milk. So where around the world are there lactase hotspots? And here's a map of the old world where the dark blue represents areas of the old world where some 90% of the population can drink milk as adults. That is, they are lactose tolerant. They continue to make the lactase gene. And they're scattered around the world, right? There's a clump of dark blue in Northern Europe, Scandinavia, the British Isles. There's another clump in East Africa, a little tiny one in there's a bigger one in West Africa, a little tiny one in East Africa. And there are also hot spots in the Middle East and in Southeast Asia. Interesting. Because we think of dairying as occurring in many parts of the world. Surrounding these hotspots are gradual declines in the proportion of the adult population that can drink milk. So that suggests that although many people have dairy animals, relatively few proportion of the world actually can drink milk in its liquid form without getting sick. So why do some populations have a high prevalence of lactase persistence while others do not? There must be something special about lactose, because you can certainly get the nutritional benefits of milk from yogurt, right? In that case, the bacteria that made the yogurt broke down the lactose. So if you're intolerant, that should be no problem. Similarly with cheese. So why is it that some populations drink milk in its liquid form and others don't? Well, there are a number of hypotheses, and we don't quite know which is the correct explanation. One is that the sugar in lactose provides calories. Well, we can also get the calories from yogurt or other or cheese. The sugar in lactose in liquid milk is available immediately. You don't have to process anything. The milk has a protein called lactoferrin that's an antibacterial. So there may be some advantage to that. Milk provides a clean source of water, which may have been beneficial in certain areas. And milk may enhance the absorption of other nutrients, such as calcium. So we have all of these hypotheses about what would the benefits be of drinking liquid milk. And we're not certain which one is correct. We do know that there is more than one way to drink your milk. Among Europeans, you saw there's a chunk of people in Northern Europe who can have the gene to make lactase. There's one mutation in Europe for the continued synthesis of lactase. That particular variant in lactase is not found in Africans. So when we first went to East Africa, where we knew people could drink milk, and we didn't find that variant, we were very puzzled. With the advent of genomics, we now know that the same ability to digest lactose and to continue to make the enzyme is accounted for in East Africans by a different variant at that same locus. So we have at least 3 variants in the lactase locus. So to summarize, Human variation is abundant. It's distributed around the world in patterns that are determined by history, such as migrations, history such as your ancestry, and your individual life history, and your adaptations to the environment of your ancestors. So a point to consider is that human biological variation, as I've demonstrated, does not form categories. We can observe the variation, we can explain it, and it is what it is. Variation is just that, variation. And we can try to figure out where it came from and why. And I'd be happy to answer more questions. Thank you.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.